Hello there. So this is my attempt at measuring the stomata, and then I'm going to go through the test that was associated. I did 19 samples because it said that I had to decide how many to do. So I set off to do 15. So I did 15. Remember, sample 1 isn't area 1. Sample 1 corresponded to a random number. So I got 19, and I'd done these quite quickly, to be honest. So again, if you've done the same as me, you may disagree, and you're probably right because I didn't do it accurately. I kind of just did it glancing at a screen. If you kind of double checked and things, then it would be better. Anyway, I've also calculated a running mean. So for example, I put 16.0 here because 19 plus 13 is, what is it? Well, 32. 32 divided by 2 is 16. So 15.7 times 3 is the sum of these, and so on and so on and so on. But I got to 15 and I got 13.1 twice, but it was like heading upwards. It's gone 12.7, 12.8, 13.1, 13.1. So I did another one to confirm it was 13.1, but it carried on going up. So it carried on going up, carried on going up, carried on going up, and then I stopped when it came down again. It's quite subjective when to stop on a running mean. The more you do, the better. But if you just did 3, it wouldn't be suitable for this kind of investigation. You should be doing at least 15, and quite frankly, in exam, I'd say do 20, and then examine the running mean. Don't say examine the running mean first, because then you might have stopped at number 8. It's gone 13.1, 12.7, back to 13.1. All right, I'll stop there, or I'll stop at 12.9. Yeah, but yeah, crucially, I've carried on using the running mean method. Odds are they'll ask about that. So we're going to look through the questions that they asked. Again, you used to have to do that process where you do a lesson collecting data, then you draw graphs of it. I've missed that step out just for ease. And then you sit an exam in exam conditions, all together as a year group, and they ask common sense questions. And now all they've done is put these common sense questions into our final exams. So there's not a separate skills exam. It's one big exam for us guys. Well, sorry for you guys, not me. I still do it. I just do it, unofficially. Anyway, so it says here, where are we? Yeah, so these questions relate to the investigation. So we need to pretend that we've done this peeling a leaf, put it under a microscope, focusing on the lower epidermis. It says use your task sheet. So that's what had the method on. Your results and graph to answer the questions. Well, you've got your results. We are bypassing the graph bit. We'll see how much that we can do. It says you were given 3 leaves from the same plant. Suggest one reason why the leaves should be from the same plant. Well, because the difference between individuals is the alleles of genes. Okay, so I just put they are genetically identical. That stems from that our phenotypes are due to the environment and our genotype, and so therefore I can give an environment answer saying the plant has been grown in the same environment or same conditions, or I can give a genetic answer. Any will do. It's a suggest question, and there'll be lots of suggest questions in this. Number 2, you were told to decide how many fields of view to use to count the number of stomata. Explain how you decided how many fields of view to use. Well, we've already done that. So we looked whether running mean stabilized. Oops, pen's got a bit funny. Yeah. Oops, I missed that mean. Sorry, running mean. Number 3, you were told to count the number of stomata in the field of view using the highest power on the microscope. The magnification depends on the eyepiece lens and the objective lens. Describe how you'd find the magnification used. You would multiply those two. Multiply them. Yeah, the eyepiece lens is times 10 normally. The objective lens would be times 40, therefore times 400. Yeah, it's only worth 1 mark. I don't need to get graticules out and things like that. Measure the field of view. It's really the thing to take away from these kind of questions is that they tend to be obvious answers. Number 4, here we go. Give one precaution you took when counting a number of stomata in a field of view to make sure the count was accurate. Yeah, well, I would have double checked, double count to see if answer is the same. That's all we need to do. Or we could have talked about that when half in, I counted as in. Yeah, be like the top left rule in ecology. They allow use of a tally as well. Use of a tally. Anything that means that what you count equals how many is there. Number 5, you were told to count the number of stomata in different fields of view of the same leaf. Explain why this is important. Well, if we only counted one area, that might not be representative. Okay, so the number of stomata per area may vary. Yeah, so it's to gain results. That's supposed to be that, or representative. That's the word to use of the whole leaf. Yeah, I could say, the stomata are spread unevenly on the leaf, and therefore I want to get a measure that represents the entire leaf. Saying the same thing. It's only worth 1 mark. I've given 2 there, so there's a simple, one version, or yes, each. I don't have to write all of that for 1 mark. Either or have my 2 little bullet points type stroke things there. So use the data to you obtain in your investigation. What can you conclude about the number of stomata per field of view in different leaves of the same plant? Can't do that because I didn't give you any data. So ignore question number 6. I think I told you to miss that out on the questions anyway. Number 7. Or if we did have it, we'd have mean and standard deviation, and we'd talk about significantly more or overlap of standard deviation. Anyway, 7. A student carried out an investigation similar to yours. In her investigation, she found out the area of the field of view and then estimated the number of stomata in the lower epidermis of one leaf. Describe a method she could have used to find the area of the field of view. So yeah, we've done this. Yeah. So yeah, so we oops, pen. There you go. So use a stage micrometer. Yeah, to find diameter. And use pi r squared to find area. You could even use graph paper and count squares if you wanted to, but not that A-level-y. That's the best A-level answer. 7b, describe how she could have used her calculation of the area of the field of view when estimating how many stomata were in the lower epidermis of one leaf. So we're going to need to know the leaf area, aren't we? So we need to draw around leaf on graph paper. And then calculate area of leaf from that. I could say how to do that if I'm running out, if I haven't got 4 marks in a minute. Then I'm going to do the calculation of so it is so leaf area divided by the area field of view times by a mean number of stomata per field of view. So that there gets 3 marks because I've mentioned leaf area. I've divided it by the area of the field of view, like you do with a quadrant in a field and things like that, and then times it by the mean number, gets a mark. So the only extra mark that I need to say is how I work out the area of a leaf. You have to draw around irregular things on graph paper. Section A done, I think. Oh no, nearly section A done. Number 8, in the investigation, the student compared leaves from xerophytic plants with a plant similar to one that you use. She found that the leaves of xerophytic plants had a lower number of stomata. Explain why having fewer stomata would be a benefit to the xerophytic plant. So this is an explain question. This is where I need to bring in some A-level biology. Explain. So yeah, there'd be less diffusion. I mean, let's use the word less transpiration. That's the diffusion of water from a plant due to evaporation. Less transpiration. More water retained. And more water retained because the benefit is that they live in dry places, don't they? Because they live in low rainfall, arid or dry conditions. How easy is that second mark? And that's the mark that a lot of people miss out. Is that the benefit of having fewer stomata to the xerophytic plant is that they can survive in low rainfall or dry conditions. Okay, and the explain bit is transpiration. So that's section A done. Really easy, but it's only easy when you know how, or when you see the answer, you're thinking, oh, is that all you have to put? And that's why you have to keep practicing these kind of questions, because you only get better by practice. So then we have a resource sheet for section B. Introduction: environmental factors can affect the density of stomata and the lower epidermis of leaves of plants of the same species. Right, so how close together the stomata, or how many stomata per leaf, is related to the environment. So the scientists investigated how growing plants at different temperatures affected the density of stomata, lower epidermis. They grew plants of the same species from seeds. Their methods are outlined below. It took 8 days containing soil, 50 plantlets in each tray. They put each tray in controlled environment at set temperatures. The plants are grown from seeds. They selected 20 fully grown leaves from each plant. They determined the mean number of stomata, like we did. Good. The results are shown here. So the temperature at which the plants were grown, mean number of stomata. When they're grown in cold conditions, they have lots of stomata. Then it decreases. Then it seems to not do a lot between 12 and 20 degrees C. Okay, so I've taken in resource A. Let's have a look at the questions that refer to resource A first, and then we'll get back to B. So it says, give 3 environmental variables other than temperature that the scientists would have to control when growing plants. Well, what things affect plants is all we need to think of. So let's go for, light for photosynthesis, isn't it? Light intensity. Light's a bit vague. I always like to put light intensity. They also need CO2 concentration. And we have to control water, don't we? So let's put water in soil. Water in soil. I can't say water, carbon dioxide. I need to say things. Yeah, but I could go water in soil or humidity of air. In fact, they can be separate marking points. There's lots I can go for, but seeing as photosynthesis is, I need light, carbon dioxide, and water, I may as well put the 3 things that it needs for photosynthesis. They also allow type of soil, a bit vague, but they allow it. Mineral ions, or in an example like nitrates, pH of soil. If you ever stuck in biology, pH, bring it down. Number 10, scientists use a range of temperatures from 6 to 20. Use the data to explain why they did not use temperatures above 20 degrees C. Why did they choose not to go above 20? Well, there's no significant change. It's just wobbling around, isn't it? There's no change. It's already leveled out. Okay. So I'll just say that. Well, yeah, that's how they've worded it. It's already leveled out. For 20 degrees C. Well, I could say it's pointless doing more than 20 degrees C because there wouldn't be any further change. The scientists only selected fully grown leaves from the plant. Select why? Well, it's just a control variable, isn't it? Yeah. So Why did they not use younger leaves? Well, I can just say that. Well, younger leaves may have different stomatal density. Yeah, it's just a control factor. Yeah, it might change as they get older. The number of stomata might change as leaves age. I could say that. Okay. You see, it's like, oh, it's quite hard to know what to put there. But when you hear the answer, it's like, oh, is that all you have to put? Yes, that's all you have to put. Again, it's why practicing these kind of questions is useful. I think you get confident at them. Number 12, and these kind of questions are done appallingly in the exam. If you can get half marks on these kind of questions, A grade, trust me, A grade, half marks on this kind of stuff. Yeah, 60% will get you an A star. So yeah, the plants grown at high temperature had lower numbers of stomata. Yes, we said that, didn't we? This would be an advantage to plants because the transpiration rate increases as the temperature increases. Explain. Oh, good. I do like an explain question because that means I can bring in my knowledge. Explain why the transpiration rate increases when the temperature increases. So anything to do with temperature. So higher temperature, higher temp equals greater. I would have written greater there. Kinetic energy. So there's faster diffusion of water. That'll do. I think what else I could put. I mean, you could say that warmer air can hold more moisture, therefore there's a steeper concentration gradient. But quite frankly, those two are. Let's keep it simple. Right, resource B now. It says use resource B. So let's go to resource B and have a look at resource B. Again, the good thing is that in our exams, all these are like the data is where you need them. It's not as if you have to keep going back to this resource sheet, which is a bit of a faff. So scientists use fossil fuels from one species. So hang on, scientists used fossil leaves. I can't even read. Sorry, fossil leaves from one species of pine tree to investigate whether changes in the concentration of CO2 in the air over long periods of time have led to changes in the number of stomata in the leaves. The method is here. They selected sites of different age. They collected 11 to 24 fossil leaves from each site. They found the mean number of stomata on the leaves per millimetre squared. They estimated the age of the sample by dating organic remains around the leaves at each site. They compared the results of the fossil leaves with leaves of the same species of pine tree growing there today. They knew the concentration of CO2 in the air at different times in the past, from ice core data normally, and the results are shown below. So present day, getting further and further back in time, 30,000 years. Concentration of CO2, right? So it's higher now. It used to be lower. It used to be higher before then as well. So it's fluctuated, but that's neither here nor there. And the mean number of stomata. So 92, 87. What I like to do on these numbers is just highlight max and min. So where's my highest number here? Highest number is there. Lowest number is there. Okay, highest number here is there. Lowest number is there. Just to kind of make sure that I don't say something stupid later on. Oh, and plus or minus is showing standard deviation. Right. Always make a note of that because it can show different things, can the plus or minus, as you'll discover as you do more stats in year 13. So, the concentration of CO2 in the air has changed over time. Use the data to describe how. This is a describe question. I need to say how it changes over time. Describe. Yeah, so I just need to say two things about it that are not incorrect. Because I can't just say it decreases and things like that. So how has it changed? So the more recent the sample from 2,500 years ago, so it's increased from 25,000 years ago. Okay, and then let's have a look more critically at the data. So it's going 190, 188, so barely any difference there. 195, 205. So imagine plotting that data. Yeah, if I had date on the bottom, and this is the present now. So it's kind of like increasing a bit, and then it's going up steeply at the end, isn't it? It's increased a lot in that last bit. So that's the significant bit, the significant increase. So you can say that's 25,000. Yeah. So pen. So from 25,000 years ago, CO2 has increased. So I've said that, and I can say that there's a largest increase. In the last, it was 5,000 years, wasn't it? It was 5,000 years, wasn't it? Yeah, 5,000 to present day, it's increased the most. Yeah, that's good. Yeah, people very rarely get 2 marks on those kind of questions. Yeah, they'll say, CO2 increases as time increases, and get it completely the wrong way around. Because time hasn't increased. It kind of has, but the data's got. It's talking about years ago into it, and the numbers are getting smaller, not bigger on that one. Anyway, the scientists calculated the mean number of stomata per millimeter squared and the standard deviation. What does standard deviation show? Again, this is bookwork. Define it. It's a measure of how spread the data is. About the mean. Spread of data, 1 mark. About the mean, 2 marks. Again, learn your definitions of standard deviation. You don't need to say that one standard deviation equals 67% of the population, whatever it is. So 15, the scientists found the age of the fossil leaves by dating the organic remains around them. Will this have affected the accuracy of their data? Explain your answer. So I can say yes, it would. Yes, it definitely would. Yes. The leaves may have moved in the organic remains, as in the organic remains are not typical of the age of them. The organic remains. Worm activity might have moved them around. Yeah, or I could say no, because unlikely to be disturbed, Therefore, age of leaf equals age of remains around it, remains next to it. Yeah, I can go for either. What a rubbish question. Don't like question 15, but you do get them like that. Would I have got that right in the exam? I would have written an answer and not been confident. But as soon as I get to explain one, I think, oh, good. I've revised. I know my stuff. 30,000 years ago, the mean number of stomata per millimetre squared on lower epidermis of the pine tree was much higher than it was today. This would have enabled the plant to grow faster when the CO2 concentrations of the air was low. So yeah, they have more stomata. So why would they grow more? Because they'd get more CO2. For photosynthesis. I know you haven't done photosynthesis yet in year 12. You'll be doing it pretty soon in year 13. But yeah, plants do photosynthesis, don't they? And they need CO2. 17, a student who saw these results concluded that as the CO2 concentration of the air had increased, the number of stomata had decreased. Do the results support this conclusion? So that's where we need to look critically at the data again. So let's just go back to it. So they've said, as the carbon dioxide concentration of the air has increased, the number of stomata in leaves has decreased. So let's have a look. Where's the data gone? Well, so there's an issue here, isn't there? So there's not a correlation there. I mean, overall there's a correlation, isn't there? Because generally that's increasing. However, there's issues here and here, isn't there, as well? Yeah, that can't. That generally, as we move towards the present day, the CO2's generally increased and the number of stomata has generally decreased. However, then I can point in my however, yeah, that there's not been a great, it's kind of, it's increased here, but it's kind of, it's gone lower, 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 then it's increased again now, isn't it? So it's increased in the last 5,000 years. What other information did they give us as well? Let's have a look. So they collected between 11 and 24 leaves. That's not a lot, lack of data. Say leaves, we're just on one species, aren't we here? Yeah, we're on pine tree. And let's look at the standard deviation as well. So standard deviation, we can talk about the significance of standard deviation as well. Okay, you know, that there's no overlap, is there, between 1,500 and 10,000 years ago? Yeah, there's no overlap there. So that is significant. So let's put all that together. Again, I only need to make 4 marks. Well, that's the kind of thing that you need to do. Just look critically at all the information. So let's get the easy one here. So yeah, they just said of leaves. It's like they're on the news, just making a blanket statement. Yeah. So first of all, let's say there's only a small sample size. There's 8 to so many leaves. So that's crucially, yeah, and only one species studied. Pine leaves studied. Okay. Overall, there is a negative correlation. Let's put i.e. less CO2 equals more stomata. What else can we say? Oh yeah, there's again, I'm not going to go back to it, but point out inconsistencies in data. Inconsistencies in data, in that generally there's a negative correlation. However, and I don't want to zoom back to it now, but there was issues between these concentration increased between these times, and yet it didn't do what we would expect. And then refer to standard deviation somewhere. Yeah, point out how it either supports the conclusion or doesn't support the conclusion. Okay, only need 3 of them. I've put 1, 2, 3, 4, 5 down. So how easy is it when you think small sample size, only one species, and overall there's a negative correlation? 3 marks. The rest are trickier. But yeah, don't forget that if it asks you to look back to the data, you can look back to everything. And then 18, lastly, the leaves of plants that grow in dry areas usually have low numbers of stomata. Use your knowledge of leaf structure. Hooray, knowledge again. I can use what I know to suggest 3 other adaptations that the leaves might have that enable them to grow well in dry conditions. So yeah, I always think sunken stomata, don't they? Sunken stomata. Remember they have that boundary layer. They have hairy leaves, and they have a thicker waxy cuticle. I should have put hairier, really. Oh no, they allow hairy leaves. So yeah, they've got thicker waxy cuticle. Yep, that gets a mark. Hairy leaves. Yep. Sunken stomata. Yep. They also allow rolled leaves. And they also allow small surface area, but it's really the same thing. Smaller SA. Again, it's a sad day when you don't mention respiration and surface area in biology. And at least we've got surface area in. And by saying that, I've also got respiration in, but it didn't crop up in this. Okay, hopefully that was useful. Half an hour. Oof.